Hello, my name is Andy Lee, and today we will be exploring the origins of sickle cell anemia and why it has remained in certain regions. So, what is sickle cell anemia and what causes it? Simply, sickle cell anemia is when the red blood cells become deformed and sickled due to a mutation in a gene that codes for the hemoglobin. However, before we get deeper into this topic, I would like to begin by explaining the molecular biology behind this and how it plays a crucial role in the development of this disease. To begin with, we will be looking at what proteins are and how they are an important factor to sickle cell anemia. Essentially, proteins are the biological macromolecules of life, along with carbohydrates, lipids, and nucleic acids. They are formed by a chain of amino acids where one monomer consists of a central carbon atom with an amino group, carboxyl group, and a R group which determines the type of amino acid. Essential amino acids are those that our own bodies cannot synthesize on their own. Thus, we get them from our foods. These include histidine, lysine, threonine, isoleucine, leucine, methionine, tryptophan, phenylalanine, and valine. Proteins play various roles in our body. Our hair and nails are formed by a protein called alpha-keratin. In our blood, the hemoglobin protein carries oxygen throughout our body. In our brain and nerves, the ion channel proteins control brain signaling. They form enzymes which help with anabolic and catabolic reactions for metabolism. They are cellular construction workers as clusters of proteins form molecular machines to do work, such as copying genes and making new proteins. They are antibodies which help defend against viruses, bacteria, and pathogens. They are cellular messengers as they are receptor proteins that transmit signals to partner proteins. And finally, in the muscles, the actin and myosin proteins enable all muscle movement. A protein is formed by around 8 to 4,000 amino acids bonded together by what is called a peptide bond, where it is a result of anabolism by condensation, and the ribosome condenses amino acids into a dipeptide. 50 or more amino acids is called a polypeptide, and the genes and DNA code for the amino acid sequence. In the primary structure, we can see the peptide bonds between the amino acids, and this results in a linear peptide chain. In the secondary structure, we can see the hydrogen bonds between the nitrogens and oxygens, and this results in the folding of the peptide chain, which creates either an alpha helix or a beta pleated sheet. Then, in the tertiary structure, we see the three-dimensional folding pattern due to the side chain interactions. They can either be ionic bonds, hydrogen bonds, disulfide bridges, or hydrophobic interactions, and results in a precise 3D shape. Finally, in the quaternary structure, a functional protein is formed, where one or more tertiary structures combine as shown in hemoglobin. Although it may seem like a small detail, the shape is very important as its specific structure determines how the protein functions. The shape is determined by the sequence of amino acids, and in cases where there is an error such as the mutation in hemoglobin that causes sickle cell anemia, the resulting protein can become dysfunctional and potentially harmful to our health. Now that we have a thorough understanding of what proteins are, we can move on to what mutations are and how it affects sickle cell anemia. Mutations are where there are random changes in the DNA and forms new alleles. Today, we will be focusing on point mutations, which is when a single nucleotide is affected. The first is synonymous mutation. This is when the change in the base sequence has no effect on the amino acid that is translated. Next is the missense mutation, where the change in the base sequence causes one different amino acid to be translated. Finally, we have the nonsense mutation, which is when the change in the base sequence causes a complete stop and shortens the polypeptide chain altogether. Out of these 3 point mutations, the missense mutation creates sickle cell anemia as there is a base substitution in the 6th codon of the gene for beta hemoglobin, where CTC becomes CAC and valine is translated instead of glutamic acid. As mentioned before, the shape of a protein is determined by the sequence of amino acids, and in this case, Valine deforms the red blood cell due to its nonpolarity as opposed to glutamic acid's polar properties. To further elaborate on this, we must know what genes and alleles are. Genes are a heritable factor consisting of a length of DNA and influences a specific characteristic in our organisms. Alleles are various specific forms of a gene, for example, eye color. In our DNA, there is a normal allele for hemoglobin, which can be labeled with HbA, and a sickle cell allele, which is denoted with HbS. These alleles group themselves into pairs, so there are three possible outcomes: HbA, which indicates healthy red blood cells, HbS, which indicates mild anemia as the body makes both healthy and sickled red blood cells, and finally HbS, which indicates severe anemia as the body only produces sickled red blood cells. Since the red blood cells become deformed, they stick together and get trapped in the capillaries. This causes anemia due to blockages in the blood vessels, and oxygen cannot be distributed thoroughly, even though the sickled blood cells already have low oxygen carrying capacities. As well, it can cause a splenic sequestration crisis, which is when the sickled blood cells clog the blood vessels in the spleen and lowers the circulation rate. Thus, less healthy red blood cells will be in circulation. So why does it remain in certain regions? Sickle cell anemia is rare to see in regions with colder climates such as North America compared to regions with hotter climates such as Africa. This is thought to be because sickle cell disease offers immunity to malaria, which is a parasitic disease that is commonly found in tropical regions. Malaria is caused by the transmission of the Plasmodium parasites by female mosquitoes. Since these parasites latch onto the concave portion of healthy red blood cells to grow and multiply, sickled red blood cells prevent such a phenomenon, as there is no concave and makes it impossible for the parasite to attack it. Eventually, the immune system will kick in to eliminate the small population of the Plasmodium parasites, and therefore, it is difficult to be infected with malaria with sickle cell anemia. Thus, this is the most accurate evidence as of today to why sickle cell disease has remained in certain parts of the world. While it may seem like a manageable disease, it can still be very deadly in situations where the patient experiences severe anemia or leads to grave complications. So, now we have a thorough understanding of proteins, point mutations, what sickle cell anemia is, its genetics, the complications linked to this disease, and why it still exists in different regions. I hope you gained some knowledge about sickle cell anemia, and thank you so much for your time. If you have any questions, feel free to email me or leave a comment on my discussion thread. Thank you.